Now let us see the disaccharides. Disaccharides are basically categorized as one of the categories of oligosaccharides, and these are the carbohydrates which on hydrolysis, either by using an acidic medium or by simple hydrolysis, they or by enzymatic activities, gives 2 units of monosaccharides or 2 monosaccharides. The commonly seen disaccharides we have is sucrose, maltose and lactose. Let us see first sucrose. Now what we need to know is that the sucrose is a disaccharide of glucose and fructose. It's a disaccharide of alpha glucose and beta fructose. Now this is the alpha glucose I have drawn, and this is the beta glucose. The OH group on the second carbon is on the upper side. Now this OH of the carbon number 1 of the glucose and the OH group of the carbon number 2 of the fructose, they both joins each other and forms this ether linkage. So this ether linkage is called glycosidic bond, and this is the bond which is actually mainly responsible for joining the 2 monosaccharides. So monosaccharides and carbohydrates are joined to each other through a glycosidic bond chemically, which is an ether linkage. Now this is how we get sucrose. Now sucrose, we have an important term related with sucrose, that's called invert sugar or inversion of sugar. Sucrose, remember, is dextrorotatory. So sucrose as such is plus. When added to water, this on hydrolysis gives glucose and fructose. Glucose is having plus 52.5 degree optical rotation and fructose is having minus 92.4 degree optical rotation. As a result of which the mixture of the two becomes levorotatory. So this conversion or the inversion of the optical rotation from plus to minus on hydrolysis is called inversion of sugar. Now let's move to second category of the disaccharide, maltose. Maltose is a disaccharide of the 2 alpha D-glucose molecular species. I have drawn 2 alpha D-glucose molecular species, and what happens is the 1 glucose molecule's OH group of the carbon number 1 combines with the OH group of the 4th carbon atom of the another glucose molecule, and these 2 OH groups, since on the same side, joins to each other through this kind of bond, which is called glycosidic bond. This is also called C1-C4 bond because C1 of the 1 glucose molecule is bonded to the C4 of the other. So remember, long chains of the carbohydrates are formed mainly by the C1-C4 linkages, or maybe due to the C1-C2 sort of linkages. But mainly when we are having glucose chains, it's due to C1-C4 linkages. And cross linkages are formed when the 6th carbon OH group combines with the 1st carbon glucose of the other. So that is called C1-C6 bond. But again, the nature of both of these, whether it is C1-C4 or C1-C6, is ether, and they are called glycosidic bonds. The third example of the disaccharide is lactose, which is the disaccharide of beta galactose and beta glucose. Now galactose and glucose only differs on the 4th carbon atom. Its OH is again on the left hand side on the 4th carbon atom, therefore drawn upward. When this beta galactose combines with beta glucose, since its carbon number 1's OH is on the upper side. Now the 4th carbon's OH and the 1st carbon OH of the galactose and glucose, they both combines with each other through this kind of a linkage, which is called again glycosidic bond or glycosidic linkage or C1-C4 bond. So this is how we get disaccharides.